Good afternoon, everyone. Welcome to today's webinar, Delivering Therapeutic Proteins into Cells Using Biological Nanoparticles. I'm Jonathan Weissman, member here at Whitehead Institute, where, among other things, my lab develops the type of therapeutic proteins that Aditya's work is helping to deliver to disease in normal tissue. I'm excited to be your host and moderator today. Today's webinar is part of a series of talks that we've organized to share Whitehead Institute science and research with the wider community. In a few minutes, we'll hear from our presenter, Whitehead Institute Fellow Aditya Raguram. First, I would like to let you know that this webinar is being recorded for future viewing. Also, we'll be answering questions at the end of today's presentation, but you can submit a question at any time using the Q&A box at the bottom of your screen. With that, I'd like to introduce our speaker today, Aditya Raguram. Aditya began his scientific career at Harvard College, where he earned an AB in chemistry and physics in 2018. His undergraduate research spanned molecular, chemical, and computational biology at the Singapore-MIT Alliance for Research and Technology in Singapore, the MRC Laboratory of Molecular Biology in Cambridge, and the Broad Institute of MIT and Harvard. In 2023, Aditya went on to earn a PhD in chemical biology from Harvard University under the mentorship of Professor David Liu, before joining Whitehead Institute as a fellow later that year. At Whitehead Institute, Aditya investigates how to deliver therapeutic molecules into cells within the body. Specifically, he's exploring the questions, how are large molecules like proteins and RNAs transferred between cells, and how can we use these transport processes to deliver therapeutic molecules into disease cells? Today, Aditya will discuss how his lab develops biological nanoparticles that can safely and efficiently deliver therapeutic proteins into cells within the body. And he'll describe how these delivery technologies could enable new therapies for genetic disorders, cancer, and other diseases. Aditya, welcome and thank you.
Thank you, Jonathan. Let me share my screen. All right, does that look good? Okay, great. So thank you, Jonathan, and thanks all of you for tuning into this webinar today. I'm excited to share a bit about our work and tell you about how we're using some of that work as a foundation for our current research that we're getting off the ground now in my lab, which we started a few months ago here at Whitehead Institute. So a large portion of our work is motivated by the promise of therapeutic genome editing and this revolution in modern genome editing technologies that provide enormous potential therapeutic capabilities. And so over the last decade, numerous labs around the world have contributed significant advances in our ability to specifically modify the sequence of genetic material, DNA, and also epigenetic material, so how DNA is expressed in cells. And as just one example of the power of these technologies, if a disease-causing mutation is present in genomic DNA, for example, a mutation that could cause a disease like sickle cell disease or progeria, that disease-causing mutation, now with modern genome editing technologies, we could design a molecular machine to specifically correct that mutation and revert that mutation back to the correct DNA sequence. And in doing so, that really results in a potentially transformative therapeutic change where the original mutated sequence is converted again back to a healthy copy of the DNA sequence. In addition to correcting pathogenic disease-causing mutations, we can also use genome editing tools to install different changes in genomic DNA throughout the genome. And because DNA provides the instructions for many cellular processes, the ability to site-specifically modify different DNA sequences can allow us to reprogram cells to have new therapeutic functions. And so the power of genome editing really has the potential to transform the way that we treat genetic diseases and other types of diseases that affect human health. And so when we think about correcting genetic mutations or making changes in DNA, we really have to consider the context in which we are doing so. That DNA that's present in the genome of a human cell is located within the nucleus of that cell. And so in order to actually change that specific DNA sequence, we need to have some way of introducing our genome editing agent or genome editing protein inside cells, and specifically inside the nucleus of cells, so that that genome editing protein can access the DNA it needs to make a therapeutic DNA change. And so this problem of introducing gene editing proteins or delivering these proteins into the cells where they need to go in order to have a therapeutic effect is really a challenge that prevents the broad application of genome editing tools to treat different human diseases. And this challenge is really more broadly characterized as the so-called protein delivery problem. And so cells in many ways are like a home with a built-in security system that generally prevents outsiders from entering the cell. So if we are trying to deliver a foreign protein like a genome editing agent into a cell in order to affect a particular genome edit, cells are generally resistant to having foreign objects introduced into them. And this resistance is especially true for large molecules like proteins and genome editing agents, which many are thousands of times larger than typical small molecule drugs like aspirin or ibuprofen, which can get into cells through much simpler processes and don't face as many stringent barriers to intracellular delivery. And so the problem of protein delivery is general in terms of affecting any type of protein therapeutic that we might want to deliver into a cell, such as a protein that needs to operate on some sort of intracellular target to exert a therapeutic function, of which genome editing and epigenome editing agents are two important examples. So what we need are delivery methods to actually overcome this protein delivery problem and introduce therapeutic proteins of our choosing into diseased cells. And specifically, the question that my lab is really investigating is not only how can we deliver proteins into cells in culture, because when we grow cells in the lab and a petri dish, it turns out that protein delivery in many in vitro systems, which still use living human cells, but these cells are kind of cultured outside of the body. These in vitro systems are amenable to protein delivery through a variety of established techniques like electroporation or transfection. But the ability to actually deliver proteins into cells within the body or in vivo, and that is within the body of an organism like a mouse or eventually potentially a human patient, those sorts of applications are some of the most impactful therapeutically, where if we had an efficient and safe protein delivery modality to actually introduce these therapeutic proteins into different cell types within the body, we could then treat genetic diseases or other diseases using a simple injection approach to introduce these proteins into the correct cell types where they need to go. And so the question of how can we efficiently deliver proteins into cells within the body is really the central question that my lab is working to answer. And we develop new delivery technologies to achieve this goal and enable new therapeutic approaches. So what are some of the challenges that we need to overcome when actually delivering therapeutic proteins into cells and designing new sorts of delivery vehicles or delivery modalities? Well, let's start by considering a general protein delivery vehicle and the path that a therapeutic protein needs to go through to actually make it from the outside of a cell to the inside. And in doing so, these proteins must cross several biological barriers to efficient delivery. And so an ideal delivery vehicle must package and encapsulate its protein cargo to protect it from degradation. prior to encountering target cells for delivery. And an ideal vehicle might specifically engage target cell types by binding to cell surface receptors that are expressed on the surface of those cells that you would like to deliver to. And upon receptor binding, these vehicles might be able to enter cells through a process called endocytosis, thus entering what's called an endosome, or a membrane enveloped compartment inside the cell. But making it into endosomes is not enough, because once the delivery vehicle, once it's inside, actually endosomes are an example of the kind of biological security system that cells have to take up foreign molecules and degrade them inside endosomes before they can actually escape into the inside of a cell. But in our case, we want the delivery vehicle carrying a therapeutic cargo to be able to escape an endosome and release its therapeutic cargo into the appropriate subcellular compartment. And so all of these challenges to biological delivery must be overcome in order to efficiently deliver a therapeutic protein where it needs to go inside a cell. And decades of research by many labs in bioengineering, chemical engineering, have tried to develop synthetic materials like nanoparticles that can very cleverly overcome some of these barriers to molecular delivery by encapsulating a cargo and then uncoating in the process of endocytosis and escaping the endosome. And many of these approaches have been successful in some cases. However, in general, it is difficult to engineer from the bottom up a new synthetic system that can actually overcome these pretty challenging biological situations. But luckily for us, nature has already given us a solution to the delivery problem, which are viruses. And so viruses have naturally evolved to overcome the challenges that typically keep large molecules outside of cells. But viruses, of course, are able to sneak inside cells and deliver viral genetic material. In the context of a natural virus, that material will go on to replicate and produce more virus. And viruses have evolved potent mechanisms to bind receptors on the surface of target cells, enter cells through endocytosis, and then importantly to escape endosomes or fuse with endosomes to actually release the viral genetic material inside the infected cells. And here is just one example. I'm showing adeno-associated virus, or AAV, which is an FDA-approved delivery vector for gene therapies. And so how might we be able to use a virus or viral vector to deliver a genome editing agent into cells? Well, we could replace the viral genetic material with a DNA that encodes the expression of a base editor or other genome editing agent. And then that delivered DNA will be transcribed into mRNA, which can be translated into genome editing proteins that are now present inside the infected cell. And so this strategy of replacing a viral genetic material with a therapeutic transgene in order to express that particular protein inside the transduced cells is a very powerful strategy for using viral vectors to actually deliver therapeutic proteins. One downside of using viruses for protein delivery, for delivering genome editing agents in particular, is the fact that, as I mentioned, if you deliver DNA that encodes the expression of the genome editing agent, that DNA can stick around for many weeks or months, or potentially longer in the transduced cells. And those DNA molecules give rise to many copies of mRNA, which each give rise to many copies of genome editing protein. That protein can stick around for quite a long time inside cells, which might not be ideal because the longer time of exposure to a genome editing agent, the more opportunities for off-target editing, so undesired editing at other locations in the genome that are quite different from your on-target site. So what we would really want for a genome editing delivery vehicle is the ability to transiently deliver genome editing proteins into the cells of interest. And in doing so, we bypass the DNA and mRNA steps and deliver the protein directly, which has a short lifetime in cells that's long enough to effect efficient targeted editing, but short enough such that it's rapidly degraded before the potential for off-target editing arises. And so in this way, we have essentially maintained the natural state of the cell prior to the treatment, where we have introduced the proteins transiently, which are then degraded rather than sticking around for a long time in terms of viral delivery. And so while viruses are really, really good at sneaking into cells and delivering their genetic cargo, what we really want is a way to deliver proteins directly, bypassing the need to deliver viral genomes. And so we wondered whether we could generate a new type of delivery vehicle that combines the best of both worlds. So we have the advantages of viruses. They can potently infect cells and escape and deliver their cargoes appropriately. But instead of delivering viral genomes, could we actually use viral scaffolds to package our gene editing cargo proteins directly, thereby then upon infection of these particles into the desired cell types, these particles would deliver the proteins directly rather than any DNA or RNA that encodes their expression. And in doing so, we might be able to generate a virus-like particle, or VLP, which could in principle combine the benefits of viruses, so their very efficient transduction, ability to target different cell types, with the benefits of protein delivery, transient delivery, and low off-target genome editing. And so the rest of this talk will really focus on this type of particle called a virus-like particle, which again uses viral scaffolds as vehicles for delivering specific cargo proteins. So in recent years, VLPs, or virus-like particles, have gained interest by many groups as potential candidate vehicles for delivering genome editing proteins, especially CRISPR-Cas9 proteins. I'm just highlighting a few examples from other labs here. But before our work, there were no examples of using VLPs for really efficient in vivo delivery, so the ability to actually deliver these particles and cargoes into cells within the body of an animal like a mouse. And again, that is kind of the real goal of a new delivery vehicle, is to show safe and efficient delivery to cells within the body. And so a few years ago, we became very interested in trying to develop virus-like particles that can efficiently deliver gene editing proteins to cells within the body, and therefore unlock the potential benefits of such a delivery strategy. And so at the time, I teamed up with a postdoc, Samaga Banskota, and we worked in close collaboration to develop this new virus-like particle delivery platform. And we were both members of Professor David Liu's lab at the Broad Institute when we initiated this work. So let me tell you a bit about how the structure of VLPs affects their function and how we engineered these particles to package specific proteins of our choosing. So in this diagram, the red proteins represent the viral capsid proteins that form a spherical shell that encapsulates, normally it encapsulates the viral genome, but we simply deleted the instructions for producing any sort of viral genome and instead replaced it with a special fusion protein, where the capsid protein is fused now to a base editor cargo protein. A base editor is a type of genome editing agent that can make single nucleotide conversions in genomic DNA. So we fuse that base editor to the capsid protein via a linker sequence that can be cleaved by a specific other type of protein. So that linker sequence first that exists between the capsid and base editor cargo allows the cargo to simply get pulled inside the particles as they're forming. And so that linker is sufficient to load and co-localize cargo molecules into the particles. But that linker sequence actually needs to be cleaved after the particles form so that the cargo can be released inside the cells that are transduced with these particles. And that cleavage is actually performed by a protein called the viral protease, which is encoded as part of the viral polyprotein shown in purple, that's also co-packaged inside the particles. So that red capsid layer is surrounded by a lipid envelope, which contains envelope glycoproteins shown in yellow. And those envelope glycoproteins determine the types of cells that these particles can actually infect and deliver cargo to. And so I like to think of these particles in many ways as biological nanoparticles, these spherical particles that package now a cargo of our choosing using proteins derived from either viruses or also cellular proteins. And importantly, the way that we make these particles is also using engineered human cells in culture. And so we provide these cells, producer cells, with instructions for making each component of the viral architecture. And that's done through plasmid transfection. And then after providing the cells with those instructions, the cells spontaneously produce these particles and release them into their cell culture media, from where they can be harvested and used to transduce new target cells. And so when these particles, we take them and mix them with cells that we want to infect in vitro. Again, there's no viral genome. The virus is not replicating at all. It's not delivering any viral genetic material. It's only delivering our base editor protein cargo, which is designed to make a very specific change in the DNA in the transduced target cells. And so we can quantify the virus-like particle potency by DNA sequencing. So when we first developed these VLPs, or version 1, V1 VLPs, we were encouraged to see that they were able to efficiently deliver base editor cargoes into cells in cell culture. But when we were trying to test these version 1 VLPs in mice by injecting them intravenously, we observed very little liver editing, around 1.5%, again, which was consistent with previous reports of, simply fusing a cargo to a capsid protein and producing these particles was not really efficient enough to yield a potent in vivo protein delivery. And so to overcome this challenge, we embarked on a campaign to systematically engineer virus-like particles to improve their potency. And along the way, we encountered several bottlenecks that we found in limited virus-like particle potency. And we engineered solutions to each of those bottlenecks to actually yield a very potent engineered virus-like particle, or EVLP. And so from our V1 VLP, we generated V2 VLPs by improving the efficiency of cargo release and kind of the efficiency at which that cleavable linker is actually cleaved to release the cargo into the transduced cells. We also, in our V3 EVLPs, performed some molecular tricks to modify the architecture to enable more efficient cargo loading in the producer cells as the particles are formed. And finally, we kind of optimized the ratio of different components of the structure to balance the amount of cargo versus other viral structural proteins that are packaged. And to summarize all of those engineering efforts, we started with our V1 virus-like particles shown in black here, and their in vitro potency, where the x-axis of this graph shows the dose of EVLP that we applied to cells in culture. And the y-axis shows the percent base editing as a proxy for the delivery potency or efficiency of these particles. And so you can see the delivery efficiency increases with increasing doses. And as we go to our V2 particles, we see the curve shift to the left, indicating an increase in potency. And with V3 and V4, we can see even greater increases in particle potency. And one way I like to think about this increase is to consider the maximal editing that we observed at the max dose of our V1 particles in black, which was around 75%. But now we are able to achieve that same level of editing at using about 1 microliter of our V4 highly engineered EVLPs, which is a 50-fold lower dose than what we started with for our V1 particles. And so we were optimistic that with this more potent architecture, we could now achieve efficient in vivo delivery using these EVLPs. So to test that, we took our V4 EVLPs and injected them into mice and investigated their potential for achieving efficient liver editing. So I'm showing right now our original disappointing data with V1 VLPs. But now with the V4 EVLPs administered at the same exact dose, we could see about 40% editing in the mouse liver, which was a 26-fold improvement compared to our V1 design. And we could increase the dose slightly and observe up to 60% editing in the bulk mouse liver with our V4 EVLPs, which is quite comparable to the editing we can achieve using AAV or adeno-associated virus delivery of the identical base editor construct shown as the green bar here. And so our EVLPs are really able to achieve as efficient editing as a state-of-the-art viral delivery method for genome editing agents. So this was promising, and we were encouraged that our new EVLP system was as efficient as viral delivery. But what would be the reason to use EVLPs over this AAV delivery if they just had the same on-target efficiency? Well, where EVLPs really shine relative to viral delivery methods is in their reduced off-target editing that comes from the fact that they transiently deliver base editing proteins. And so when we looked at in vivo specificity and detected off-target editing at several sites in the genome that are known kind of sites of unwanted editing aside from the one particular on-target site, we observed that at several of these sites, we could detect off-target editing above background levels in the AAV-treated mice, shown in green. But in the EVLP-treated mice in blue, we observed no off-target editing above the untreated mice, shown in gray, above background levels. And so these results really highlight the utility of EVLPs and the benefits of using viruses to package proteins directly, bypassing the need to deliver DNA or mRNA. And so finally, we also applied our new EVLP technology to treat a mouse model of genetic blindness. Again, the ultimate goal of using any sort of delivery technology is to demonstrate that you can deliver a therapeutic protein at sufficient quantities to actually treat a disease, in our case, starting in a mouse model. And so we collaborated with Professor Chris Palczewski's lab at UC Irvine, and his lab are experts in inherited retinal diseases and have these mice that have a premature stop codon in R44 to stop mutation in the RPE65 gene, which leads to a near complete loss of visual function. And so it turns out the stop codon can be corrected using a base editor designed to specifically convert that mutation back to the normal arginine codon. And so we produced EVLPs packaging a base editor designed to correct this mutation, and the Palczewski lab members injected these VLPs subretinally into the mice with the genetic blindness mutation. And we observed about 12% correction of this target base in the EVLP-treated mice in blue. And this level of correction, in this particular experiment, we compared to a different type of viral delivery, lentiviral delivery, again, using the same exact base editor construct. And the viral delivery in red resulted in the same exact on-target editing in blue from the EVLPs. And this amount of editing and stop codon correction led to a partial restoration, but an improvement in visual function in the treated mice. Here I'm showing electroretinogram waveforms of the untreated versus treated mice. And so you can see the untreated mice have a flat response to electrical response to light in their retinal cells. But after the treatment and correction of the mutation that's causing the blindness, we can see a partial rescue of the characteristic waveform that indicates that these mice are responding to visual light. And if we look at protein lysate from these retinal cells from the treated mice, we could see that the stop codon correction at the DNA level led to a partial rescue of RPE65 protein expression. And intriguingly, from this same Western blot data, we also observed that the base editor, in this case, an adenine base editor or ABE, that base editor protein was still detected in the treated retinal cells from the lentivirus, the LV treated mice, at 5 weeks post-injection, but not the EVLP treated mice. Again, highlighting that with viral delivery, you have prolonged expression of your editing agent, but with EVLP delivery, it's transient, and it can lead to just as efficient or potentially better rescue of disease phenotypes compared to prolonged viral delivery. So to pause for a moment and summarize are the key takeaways from our original EVLP development study. I've shown you how we engineered virus-like particles to package cargo proteins, especially base editor proteins. So we package these proteins inside a viral scaffold without any viral genome or viral genetic material present, essentially using viruses as kind of a hollowed out vehicle for delivering therapeutic proteins of our choosing to treat disease rather than causing disease like a normal virus would. Accomplishing this required several molecular engineering tricks to improve cargo release and cargo loading from the particles, starting from our version 1 or V1 architecture all the way to our V4 architecture in blue. And with collaborators, we demonstrated that our optimized EVLPs can mediate therapeutic base editing with minimal off-target editing across several organs in mice. And so we like to think of EVLPs as a new protein delivery platform that have several benefits over some existing strategies, especially viral delivery strategies. And we're grateful for this fantastic image from a talented artist, Elena Berg from the Broad Institute, who depicted our virus-like particles as these drones or spaceships carrying our Cas9 cargoes and landing on different organs like the liver and eye that we were able to deliver our cargo to in our first study. And so we really think of EVLPs as a potential next generation delivery modality that can offer key advantages and combine key advantages of existing delivery methods. And so in the last few slides, I just want to discuss some recent developments since our original EVLP report, where others have used our findings and our EVLP systems for new applications. And so in one study led by Mei-Ray An, a graduate student in David Liu's lab, she used the insights from our original EVLP development to generate new particles called prime editor EVLPs that can package a different type of genome editing agent. And this was a heroic engineering effort that required many modifications to these particles, kind of tailored to the specific requirements of prime editing systems. And this report really demonstrates the potential versatility of EVLPs of packaging different types of genome editing cargoes, from Cas9 nuclease to base editors to prime editors to potentially other types of genetic medicines. So another real advantage of using EVLPs is the ability to retarget EVLPs to different cell types simply by changing the envelope glycoprotein, or that yellow protein that's expressed on the surface of the particles, which determines the types of cells receptors that these particles can bind to and then infect. And so this particular aspect of EVLPs has really gained a lot of interest in the last few years. And in our first study, we showed that we could use a commonly used protein, VSVG, that can transduce most cell types, including Neuro2A neuroblastoma cells in blue and 3T3 fibroblasts in red. But we can also switch out that VSVG protein for a different glycoprotein, which preferentially transduces neuronal cell types. And indeed, in cell culture, we observed preferential transduction and editing of N2A, the neuroblastoma cells, compared to the fibroblasts. And so other groups have taken this programmable tropism strategy even further, including a very recent and elegant example from Jennifer Doudna's group, where they used EDVs, or enveloped delivery vehicles, which are similar to virus-like particles and incorporate some of the molecular tricks that we use to go from our V1 to V4 EVLPs. And they used these particles, and instead of a VSVG, a normal glycoprotein, they used an antibody fragment that can bind to a specific desired target receptor on the surface of a desired target cell and get cell type specific transduction using this type of envelope particle. And in their study, they also demonstrated the potential for using these particles to actually deliver a CAR, a chimeric antigen receptor, and other components into T cells within mice, thereby generating these CAR T cells using a single injection of virus-like particles or envelope delivery vehicles. And this type of in vivo CAR T generation could then be used to actually engineer these T cells to target specific cancerous cells in the body and target those cells for destruction. And so the ability to use virus-like particles or envelope delivery vehicles to specifically modify different cell populations within the body to reprogram them to have a function like killing a specific cancer cell, or maybe remodeling a cell state in some other way. The potential for these delivery vehicles to both accomplish that and also to correct genetic mutations in specific tissues that are causing disease is really quite enormous. And so we are very excited about this EVLP technology. And in my lab, some of our ongoing studies include efforts to further improve the potency of these particles through different protein engineering strategies. And we are also taking a look at how we use cells as factories for producing these particles, and whether we can gain insights into the pathways involved in particle production, and how we can exploit those insights to potentially improve the efficacy of EVLPs even further. And so I've presented one possible solution to the protein delivery problem, and how do we kind of sneak proteins through these cellular security systems using virus-like particles. But another interest of the lab is to investigate other mechanisms for delivering proteins into cells. And potentially, that can also maybe bypass these cellular security systems. And, cells are very resistant to potential intruders, but neighboring cells are able to communicate with each other just fine and send proteins or RNAs through different types of molecular transport systems. And so in this case, the cell receives specific signals that allow it to actually receive these friendly neighborly cargoes as opposed to foreign intruder cargoes. And so we are very curious about the molecular mechanisms involved in defining these processes, and whether we can leverage some of these intracellular transport mechanisms to develop different delivery strategies or improve the efficiencies and and safety of these existing delivery methods like virus-like particles. And so these sorts of studies on bioparticle biology and the overall potential for engineering these biological nanoparticles for protein delivery are the real focus of my lab studies, again, with the goal of really being able to use these particles to deliver therapeutic genetic medicines into cells in the body where they need to go in order to actually treat diseases. So with that, I would like to acknowledge a large group of people, including my lab members now at the Whitehead Institute, Diana, Hewan, and Aline, who have helped make a lot of great progress in the last few months since we've started on some of our new research directions. I would like to acknowledge my PhD advisor, David Liu, for all of his support as we started this virus-like particle project and published our paper a couple of years ago. And especially would like to thank Samaga Banskota, who worked with me very closely on the original EVLP development story. A few other LULAB members who contributed to various aspects of the virus-like particle project include Mary Ann and Jesse Davis in particular. I'd also like to thank our collaborators in the Plachevsky lab at UC Irvine, including Susie, Sam, Elliott, and Professor Chris Plachevsky, who provided support and their technical expertise for the subretinal injections that I described in using EVLPs to treat genetic blindness. And finally, at the Whitehead Institute, a few people have been especially helpful in getting our lab off the ground, including Dr. Heather Keyes, the director of the Functional Genomics Platform. And in general, the fifth floor labs are kind of our lab mates on the fifth floor where our lab is situated, as well as the Weissman lab, for being very generous in sharing equipment and resources that have been essential for us as we get started as a new lab. And I'd also like to acknowledge the sources of funding who have supported either my past work or current work that we are doing in the lab. And so thank you for listening, and I look forward to answering any questions that are available. Thank you.
Thank you, Aditya, for a fascinating seminar. So I'll take the opportunity to the first question. What do you see as sort of the key limitations you have to overcome to go to develop this as a therapeutic?
Yeah, so I think, sure. So I think probably the number one limitation now is the scalability. And so with many viral delivery methods, you require, millions of dollars and large scale production of culturing these producing cells to produce enough particles that are required to achieve efficient delivery. And so that problem still holds for these virus-like particles in particular, and because these virus-like particles are, not natural viruses or viral vectors, they have unique challenges associated with them in that, their capsid is not really optimized for doing this task of packaging large quantities of cargo proteins. And in general, production efficiencies are, potentially even worse than traditional viral vectors. And so being able to do two things: one, improve the potency of the particles on a per particle basis, which would then allow you to administer lower doses of particles to achieve the same level of editing, then reduces the quantity that you need to make, but then also to develop strategies for actually producing these particles efficiently enough to then be able to attain the levels required for efficient delivery in kind of a more feasible and scalable process. I think those are some of the main challenges for potential clinical translation.
All right, we have a question from the audience. Is it possible to target pathogenic bacteria strains as opposed to cancer cells or other cell types?
Yeah, so interesting question. So there are So, the viruses we use are, and, the envelope proteins and targeting molecules and mechanisms of cell entry are kind of more specific to human cells or mammalian cells. But there are other viruses that infect bacterial cells, bacteriophages, and there has been some work, not from me, but from other labs on using these bacteriophage-like systems or other bacterial secretion systems for targeting delivery into pathogenic bacteria. Yeah, so that's definitely an interesting area that is, somewhat related, but these particular particles that we're working with probably could not be used for bacterial delivery.
All right, we have another question. VLPs, EVLPs can transduce proteins into neural cells in vitro. Does it mean VLPs can get into the blood-brain barrier?
Yeah, so we haven't explicitly tried delivering VLPs across the blood-brain barrier. We have reason to believe that it would be challenging because most viruses cannot cross the blood-brain barrier, and especially VLPs are kind of on the larger side and are, 100 to 150 nanometers in diameter that would generally impede their diffusion through tissue and also might impede blood-brain barrier transport. So we have tried doing local injections of VLPs into cerebrospinal fluid. So either cerebroventricular injections, which are kind of analogous to intrathecal injections that are used to administer some FDA-approved gene therapies or antisense oligotherapies. So we tried these routes in mice, and we can get delivery of VLPs to brain cells when we do that in neonatal mice. The delivery efficiencies we see in brain are a bit more limited, we think, because of the diffusion of the particles due to their large size. And so maybe developing different sizes of particles, smaller particles, or again, playing with other components of the viral envelope to enhance uptake by specific cell types and kind of decrease absorption by other cell types might improve delivery in that way. But the short answer is we can achieve some brain delivery through a local injection that bypasses the blood-brain barrier.
All right, next question. Great talk, Aditya. Any thoughts on how VLPs compare to lipid nanoparticles? For example, the ones used by Verve for their clinical trials. Caveat that their first formulation had some signs of liver tox. Any indications of off-target organ tox by VLPs?
Yeah, great. So we have done some liver histopathology previously and didn't see any gross toxicity. We also looked at some liver enzymes like ALT and didn't see elevation, but we haven't done a very thorough multi-organ tox characterization in our hands. So the comparison between VLPs and lipid nanoparticles is certainly interesting and important. And so for liver delivery, especially some of the, improved LNPs, maybe the GalNAc LNPs that I think this person was alluding to, can be quite efficient for liver delivery of mRNA that encodes the expression of a base editor, for example. I think the real question and distinguishing factor between VLPs and LNPs in the coming years will be the types of non-liver organs that they are able to target. We are particularly optimistic about the retinal delivery applications of VLPs, while there are some potential uses of LNPs for retinal delivery. We think that VLPs can offer a real advantage there. And also VLPs are just different in that they can offer the opportunity to deliver ribonucleoprotein or RNP cargoes, which are historically more challenging to deliver using LNPs. And for CRISPR-based agents, RNP delivery does offer the greatest on-target editing with most minimal greatest on-target editing with most minimal off-target editing. And so those are kind of the key differences that we see between VLPs and LNPs. But we really think that different delivery vehicles might be most optimal for different situations. And for now, of course, there have been really great results using LNP mRNA in delivery, especially for liver indications.
Terrific. So you talked about the different things you can deliver, whether it's a protein RNP, or as is the case with LNPs, typically mRNA. What do you think about the possibilities of delivering other biological molecules like RNAs through VLPs?
Yeah, so we can definitely deliver mRNAs with VLPs, and there have been reports from other labs as well, using a strategy that includes the fusion of like an RNA binding protein, like an MS2 coat protein, to the capsid protein. That RNA binding protein can bind to the appropriate RNA aptamer that's present in a particular cargo mRNA that you'd like to load inside the particles. And so that could be one strategy for packaging mRNA inside the particles. There have been some reports of also packaging circular mRNAs in some similar particles as well. I think one key difference there is when you're producing these particles in cells, the mRNAs are not chemically modified, or that like they typically are for like mRNAs packaged inside LNPs, for example. So that could offer different either kinetics and a lifetime of that particular mRNA in the transduced cells, or potentially the mRNA stability as well. So those would be some differences between an mRNA packaging VLP compared to an mRNA packaging LNP. But in principle, yes, mRNAs can be efficiently packaged and delivered with VLPs. I think DNA is maybe actually a bit more challenging, although you might be analogous ways to package DNA with a DNA binding protein fused to a capsid protein, just like an RNA binding protein fused to a capsid protein could package mRNA.
Terrific. Have you investigated immune responses and the possibility of repeat administration in vivo?
Yeah, so we have not investigated repeat administration simply because in our cases, we are delivering a genome editing agent that makes a permanent change in genomic DNA. And so a one-time administration is kind of sufficient to for our purposes. And so we would expect these particles to be immunogenic because they are using viral proteins. And so it's possible that after an initial injection, even if that editing is complete, that should be sufficient for many types of applications, but might prevent the use of repeat injections. There could be other strategies that might get around the issue of immunogenicity. There's certainly some very interesting work from many labs, including Feng Zhang's lab, on using endogenous retroviruses or retroviral components that are actually found in human genomes or mammalian genomes that are potentially immune privileged, but function in a very similar way compared to these retroviral capsids and proteins that we used in our EVLPs. So these could be a way to minimize the immunogenicity of the particles, and also some of the like vesicle-based approaches that I kind of alluded to at the end of the talk might be able to, again, offer benefits to over virus-like particles in terms of immunogenicity, because they are made mostly of human protein components, mammalian protein components as well.
Have the local injections enhanced targeting and controlled biodistribution, and what's the optimal size?
Yeah, so for the local injections, that we've tried, it's mostly in, so for subretinal injection or local brain injections. And then in those cases, we definitely see increased, transduction of the desired cell type, with the local injection route. In terms of the optimal size, I think that's still an unanswered question. So as just a comparison, AAVs are smaller, around 30, 40 nanometers in diameter, as opposed to 100 to 150, possibly even a bit larger for VLPs and lentiviruses that we've observed. And so I think, if and AAVs diffuse pretty reasonably well through some tissues. And so, of course, with particle size, there's a trade-off between the the smaller the particle, the fewer cargo molecules you can package per particle, and then that you would think would decrease your potency on a per particle basis. And so that is definitely an important trade-off to consider when modulating the size of the particle. In our hands so far, we've found that getting the most bang for your buck in terms of every single transduction event delivering a large bolus of protein dose has been the most efficient. But certainly exploring that trade-off further could be quite interesting.
Terrific. Well, I think that wraps up our time here. That concludes the presentation. Thank you all for joining us today, and special thanks to Aditya for sharing your work with us. Take care.